Okay, so um, let's get back. All right. So okay. So again, this is going to be the equation, right? Uh, it's just differential equation of the first order, and the question is, we do know that in differential equation of the first order, we can have only equilibrium states, or what kind of dynamics it can produce, right? It shouldn't, because to get oscillations, you need at least two equations. So you're going to be you're going to have a system in the plane, but now you have only a single equation. Okay, and that's what mathematicians do, right? They like fooling around. So, and we need to produce something like this. You see on the right hand side, I magnified this picture just to show that you have to memorize a couple of things. First of all, in neuroscience related papers, you never create diagrams like this one, right? When you have membrane potential on the left hand side, because everybody knows that membrane should be between this low value. I mean, it can't be lower than negative 80 millivolts, right? And up to, say, 40, 50 millivolts, which basically means that if you apply a very, very negative pulse, right, like you can't apply 25 nanoamperes or milliamperes, because you're going to burn the neuron. You'll just make it dead, right? Or you can't apply too strong positive pulse, right? Because you're going to just destroy the neuron, right? And a neuron is a part of the animal, and animals, like, again, you may wonder what models they use in neuroscience, right? To do neurophysiological recordings. And it's very interesting that the animals that you perform experiments upon in wet meaning that dry versus wet lab, I typically say leeches, medical leeches, right? And one reason they use medical leeches, because medical leeches used to be uh, uh, they used to be a part of the what's I forgot, the basically Tylenol, or whenever you have a headache and blood pressure, they use medical leeches to lower the blood pressure, so the leeches can suck in some blood, right? And when Napoleon Bonaparte went to Russia to conquer Russia, right? Then they have barrels with leeches, like 500,000 leeches, and because of that, leeches were a very often used in neurophysiological studies. Almost everything is known about cellular dynamics and leeches, right? Or some kind of sea slugs. For example, here in the United States, marine biology is very popular, right? So no surprise that sea slugs or other slugs are used in experimental studies. And one reason is because they're well studied, and secondly, because they're relatively cheap, right? I do remember that uh, maybe like 15 years ago, I don't know how much is it now, but medical leeches on average was $5 per leech, right? And sea slugs are much more expensive, so you don't want to apply a pulse that can ruin the nervous system of the sea slug, because it basically means that you lost the animal, and you lost the animal. Okay, you have to pay again, right? And you must have money to pay for the experiments and for the animals. So that's why you have to be very accurate. And there's another interesting story that, for example, why nobody uses, say, cockroaches, right, to study nervous system of cockroaches, right? And one reason is because nobody wants to, because it's a messy animal, and it's very difficult to find neurons. They're small, right, and record, poke electrodes and record from small neurons, right, like in cockroaches or Drosophila flies. So there must be, again, always some kind of uh, some kind of convenience involved, right? Fish, same story, right? You can't use fish. You can't use fish because again, because to put it in on a petri dish, right? Fish must breathe in the water, right? Oxygen, but in the water, not in the air. Right? So it's going to just die very quickly. All right. So this is just an introduction. What kind of animals we use and why? Okay. So as I said, to generate action potential, so typically this is between -65, and what you see here, basically this, right? You can see voltage go slowly up, okay, in this particular case, right? Because this motor neuron it receives spikes from neurons that control locomotion, right? That control muscles. I mean, and you see it goes up, up, and up, and then it reaches a certain value around here. This is not true. This is not a rest potential. This is around, it's called voltage threshold. You see this THI voltage threshold. And this is around -45 millivolts. And then after that, cell generates action potential. And the reason it generates action potential because the gate that's specifically designed for sodium ions, it opens up only for sodium, right? And then extracellular. Then from outside of the cell, sodium ions that positively charged rushed into the cell, and then voltage goes up. It's called cell becomes quickly depolarized, which means that the sodium ions move very quickly, right? And that sodium current, is also very quick. Okay, and as soon as voltage goes to a certain level, right, the cell becomes depolarized, then the gate channel for the potassium current, for potassium ions, opens up, and the cell releases potassium ions outside, and they're positively charged, and then again voltage drops down to a certain value, and we call this repolarization. Also, it's a very quick process, right? So basically, the spike duration is roughly, you see this says 2 milliseconds. It's typically on a scale of 1 or 3 milliseconds. Okay, so that's how quick. And then this leak current basically bring the cell to the steady state. Okay, so this is the rest. And actually, it doesn't, basically, when repolarization, the voltage goes even further down than the rest potential, and then slowly the cell will recover to that level of -60 millivolts. Okay, so that's what we need, right? And this is our differential equation. Okay, and in the right hand side we either add up this term, which stands for synaptic current, that the cell gets input, whether it's a positive or negative input from other cells, or this can be just an external, or it's called applied current. So let me see what the next slide is. Okay, and this is my great handwritings. Okay, I didn't use my iPad for a long period of time, so hopefully I'll be better this time. Okay, so we look at this equation, right? Just and then this is, I'm going to use external current for time being. So let's say current is 0. So the equation looks very similar. So as I said, in neuroscience we use maximal conductance. This subscript L stands for the leak current. So that's the only current in the system. So it's G leak, right? Or again, G is a 1 over R, which is resistance. So we don't use R anymore. And this rest potential, okay, this value, which is, again, it's different from every current, okay? In this case is -65 millivolts, okay? So therefore, and I want to find what's called an equilibrium state of the system, right? So what do? Equilibrium state, when everything stabilizes, is when V prime or V dot equals 0. So then I just have this equation, right? And what I have, V prime equals 0, and then I equate the right-hand side to the 0, and I find that the steady state value is going to be basically external current plus G times what's called rest potential divided by G. So when the external current is 0, clearly it's just a rest potential, right? So you stay here, right? And this minus basically means that your corresponding differential equation solution is e to the negative this times time, right? So no surprise that this state is stable, right? It's as simple as that, because again, you just compute the derivative at this point, and you'll see that's going to be negative, right? All solutions converge to these negative 65. And what you do, okay, this is true when the current is 0, and then you add a positive current, right? And you add a positive current, so basically this 5 divided by g, then this steady state is going to move up. It's going to be not negative 65, but say negative 60 millivolts. Okay, so let me see. Okay, that's what I meant. It's the same picture. And then I apply a current like this, right? And my steady state is no longer negative 65, but the new level, this green line, which is I just shift the equilibrium state slightly up. And as long as this green line is below the spike threshold, which is roughly negative 45 millivolts, right? The cell stays silent, right? Or we say quiescent. Okay, and what we do is, again, we just increase, we add up a stronger current pulse, okay, which means you shift this green line above synaptic threshold. And what happens, and this is, again, this is our modeling approach. So therefore, solutions of this differential equation will try to get to the new target. So they're trying to chase that level. Okay, they want to converge to this new light green light, right? And what we do, we say, as soon as voltage moves up, okay, increases like in the regular cells, remember, then it generates an action potential. All we do, we just create what's called an event. We create a piecewise linear system, okay? If voltage is below V of time, is below negative 45, right? We have this differential equation, right? We just compute the solutions analytically or computationally. It doesn't really matter. But as soon as it goes, it hits that threshold, it's like a bump. We have this kind of sound, bump, bump. We just register a spike. All we do, we just reset it to a low level below negative 65 to start the game again, right? So like negative 80. And then if pulse is still there, right, then the solution will try to reach again this green line from below, and then it hits the threshold, negative 45, and then generates another spike, and then another spike. Okay, that's how we create, that's the concept of integrating fire. So we don't have spikes. We have we only register the event, and we call this a spike. So that's how we fool everyone, right? Okay, so, and what we can do, as I said, okay, we start from negative 65, reach threshold, but we will try to generate a spike and then reset it either to this level below negative 65 or even lower, or we can say even that for some period of time the cell is it doesn't talk anymore, right? It stays quiet, right? It doesn't respond to anything. You can create this kind of plateau and then start the game again, right? So you can reset it lower, so it will recover lower. And the idea is, I mean, hopefully this figure is supposed to illustrate, again, negative 65, you reset it to negative 80, and you have, so you have here black spikes. So I apply a current, okay, like this current. So if the current is below some threshold, right, so solutions don't reach the synaptic threshold, negative 45, like this one, I don't generate anything at all, right? If I increase the amplitude of the pulse, then the solution, this black solution, will try to get to this line from below, and then it gets here and generates spikes, and generates another spike. I reset it. But if I apply a stronger pulse, like 10 here, then my green bar moves up, which means this solution will move faster because it's just exponential function, right? Trying to get to the green bar, and it reaches this threshold faster, it generates action potential, I reset it, and then it chases the green line again and gets to the red level, generates spikes, reset. Basically, you should see that if I increase the current, then the number of events increases, which means it generates more spikes. Okay, and that's how it works. Okay, so this is just MATLAB code. Let me try to demonstrate how it works, right? It's supposed to be integrated file, simple file. And when you run it, you will see something like this, okay? So you start from -70. Threshold, in my case, is -20, right? Not -45. And then generate action potential point, and then reset it. And then it climbs again, reset it, climbs again, reset it, right? And then you generate this current. Okay, let me do this. I click here, go to M files, and I go to integrated file, and it says simple, right? Where is it? It says simple one. Yeah, do I have a simple one? Yeah, I have this file. And okay, so, and let me just run it, change the folder, and you should see the picture. Can you see it? Hello?
Yes.
Okay, so, all right. So when you play with this game, okay, you see these are the numbers. I integrate from 0 to 100 times, right? My initial voltage, -72, that's the rest potential, okay? This is the external current, okay? And then I guess I did it for neuroscience students, so I said try between 0 and 10. And capacitance here is 1, resistance is 10, which means that Gmax is 0.1, okay? And then we use it's called ODE solver, which is basically we instead of differential equation, we create a difference equation, and we integrate with the step dt, which is basically, what is it? It's a 0.1, right? And what I have is the right hand side. So I start from this number, which is -72, and I compute the next value, next iterate, which is V2, and I use V2 to generate V3, and so forth and so on. Okay, and then if the voltage is above starting threshold, I reset it, and it goes to the value reset. What is it? -65. Okay, so guess what? Let's try different current, right? Just follow my own instructions. When I have 0, right? Just run it, and as you can see, the solution converges to the steady state at the level of -65. Then I add, say, 2, and run it, and the solution, exponential solution, right, converges to a new level at some, who knows what it is. But my goal, my threshold is -20, and I don't reach it, right? So if I do 4, okay, nothing happens, right? Then let's add up, say 5. And as you can see, as soon as I got 5, very likely that, as you can see how the solution bends, this target is around here. It's slightly above the -20. And after that, okay, I generate action potential, reset it, generate another action potential, reset it. If I reset it, rest potential, rest. Okay, if I make it lower and make it 75, so basically I'm going to reset it right here. Now I have 4 spikes, right? You see, at this level, right, the current is not strong enough. But if I increase it, hopefully, then the cell will generate action potentials, right? So which basically means all I've done is just I delayed that climbing time, right? So it didn't have enough energy and time to reach to the above the threshold. Okay, so I go back to 65, do it again. Okay, and you see I have more spikes. I mean, like 1, 2, 3, even more. And if I make it 10, okay, if I make it 10, then you have then the neuron generates even more action potentials, right? It generates, so which means basically this means that the spike frequency in this neuron is a function of the external current over here. Okay, now if I click this file, let me just see, I don't remember, that was Kelly who did that. And let me just run this stuff. It's going to take some time. So basically, in cycle, I'm moving, increasing the external current from 1.5 is this step up to 8, whatever, nanoampere. And let me see, it shouldn't take that long, right? And I click on the figure. Okay, you can see this shows the number of spikes on the vertical axis, right? And then this is the magnitude of the external current. So as we can see before, when the current is below this particular threshold, this case is this value like 4.5, the neuron doesn't generate any action potentials. And then after that, if you increase the external current, it starts generating action potentials, right? And the number not linearly increasing, like square root of, right? As you can see, it bends like that. That's exactly what people see in experiments, right? The number of spikes is a function of external current, right? But that external current can be also synaptic input from other cells of the network, okay? So we did this part. So again, we tried integrating phi simple, and this file, Kelly integrate and phi, it shows how the number of spikes depends on external current. So I'm going to close this. Kelly, I'm going to close your file, integrate and phi, okay. And okay, now if I run this file, we're going to come back to this, right? This time, okay, as you can see, it's the same story, right? I have the current threshold is known, right? And then this voltage in time. But if I go to the plot, this, and I remove this part, right? Dot here. So now I connect the dots. Okay. So you can see I created an illusion that I have spikes. So as before, I have a climbing part. Basically, what I did is, as soon as voltage gets to this level, -20, I don't reset it, but I make a voltage 50, right, millivolts, and next step I reset it to start the game again. Okay, and now it looks like I really have spikes, right? And if I make this number say 9, clearly, then I'm going to see more spikes, right? No surprise, I'm going to see more. Okay, then the other number that is needed here, so I go back to make it 6. Okay, so I mentioned this number. It says absolute refractory period. Okay, basically 1 means little plateau here. I don't know if you see it or not. This basically means for 1 step, the voltage doesn't change. Let me make it this number 10 so you can see what happens. If I make it 10, you see this little plateau. Basically, this is called refractory period. Refractory period means that cell doesn't do anything at all, right? It stays just it stays at this rest potential, and nothing is going to happen. It doesn't respond to anything, right? If I make it 50, okay, then refractory period, you see it increases. So for 50 times, I prevent the cell from climbing again, right, and generate an action potential. So no surprise if you increase this time, then you're going to get fewer spikes than before, right? That's how you control it. Okay, and it's true. It's known that after the cell generates action potential, it needs time to recover from that spike from action potential, right? So it needs to regroup the energy to produce another spike, right? Okay, so you can control this number. Okay, just make it realistic. Make it, oops, sorry. Make it 5. Okay, again, I run it, and voila. I mean, we're going to talk about what's going on here later on. Okay, so I go back to this picture. So we discussed this. So that's a file integrating fire, right? I just put a red dot here, just saying that, oh, I have spikes, so you can see better. That's the price you pay for the simple model. Okay, but then, as I said, why do we need spikes, right? And the truth is that the spikes are needed, okay, so to tell the other cell on the network, okay, so we have now two cells, cell number one, cell number two. We call this presynaptic because you see this, there are two synapses projected from cell number one onto cell number two, so which means there is no reciprocation. So this is called presynaptic. Just get used to this jargon, right? And this is called postsynaptic, so which means it's a causality. So the signal goes from cell number one, this time into the cell number two. And the idea is, okay, so this black arm, okay, with the dot at the end means it's inhibition. And the red synapse with the triangle, it basically means that it's excitation. So the idea is the following. So whenever blue cell generate this action potential, single spike, and if you record from the other cell too, right? So you have two electrodes stuck in here and then there, you will see that that right after cell number one generates this action potential, the voltage in this cell, there's a dip here, it goes down and then it recovers. So again, in neuroscience papers, right, we don't use scales like that. We just say this negative 65, 40, right? So we put a vertical bar, like I did it here, vertical bar, and it says 5 millivolts. It's like on the map, so you know the scale. Okay, so this dip, typically, right, if it's negative 65, it's around negative 67. So which means it can't go all the way down like that, right? Because it's unrealistic. So basically it means that for you, and this is called RPSP, that echo. It's called inhibitory because the voltage goes down, which means voltage becomes in the cell 2 becomes hyperpolarized even more. It's called inhibitory postsynaptic potentiation. However, if in response to spikes in this cell, right, you will see something like this, which means there are positive bumps. We say, this cell, the voltage in the cell, the cell becomes depolarized. Voltage goes up, another like 2-3 millivolts up here, and then it recovers, right, like this. And then another spike in a blue cell results in what's called excitatory EPSP in a postsynaptic cell. Okay, that's how it works, right? Okay, then you would say, what it indicates to experimentalists is that this synapse is excitatory and that synapse is inhibitory, and we also have to learn how to model this interaction. The truth is that often it's the same synapse. Like I just did it for just an example, but it's true that happens all the times. That the synapse, it has this dot and a triangle. You know what that means? That means the synapse has two functions. First of all, there's a positive bump due to excitation, and so it's a fast excitation. Excitation is typically is fast, and then followed by this negative hyperpolarization, right? Minus, because inhibition kicks in, which means that the synapse has two functions: fast excitation and slow inhibition, like this. Okay. Now the question is, okay, this is another interesting question. How are you going to get that bump? So basically what happens is, let me just try to explain it informally. Okay, so cell number one generates extra potential. It releases messengers. They're called neurotransmitters. Okay, they're not ions. They're just special particles. Neurotransmitters, right? They released every time a spike is generated. Again, the biophysics is more complex than that. So basically, they fly towards the green cell, and then they knock on the door, tuk tuk tuk, right? Knock knock knock, and the green cell responds to this event, right? It opens up. Okay, so basically neurotransmitters, they should I tell you this? I shouldn't tell. Okay, watch the movies on YouTube, right? They make the green cell open up certain gate, right? And this open up certain gate, and for example, if I need a negative response like this, what should happen? Basically, this happens that when the gate is open, right, in response to the spike, the green cell releases positive ions, like say potassium ions, right, out. If it releases potassium ions out, that means overall it becomes more negative, right, in interior of the cell, right, which means voltage goes down. But on the other hand, when neurotransmitter reach the gate and the gate opens, it could be that negative ions from outside rush into the green cell inside, right? And then voltage goes down too. The truth is, nobody really knows this well, right? So all we can do is just register this event. How it's done, it's not fully clear, right? And it's very difficult to figure this out, okay? But the interesting part for us, right, and for you as well is, okay, cell number 1 releases these messengers, right? Okay, and what I was trying to show, for example, in this case, right, this picture on the right, you see the blue cell generates a train of bursts, or a train of spikes, right? It's called a burst. And you can see that the red cell responses, right? It goes down, wakes up, goes down, wake up. It's like an echo, like this, right? Zigzagging, which means every spike generate a kick down, right? And it generates, so you can say, the blue cell looks like the blue cell inhibits the red cell. So the connection between the cells from blue to red is inhibitory, and it's fast because it goes down with every spike, and then it goes up again, and then goes down again, right? Like boom, 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 like you like playing with the ball, okay? And so, which means I need to model this process moving from cell number 1 to cell number 2, okay? And we call this, I have to model how intense it's called neurotransmitter depletion is, the rate. And now I'm talking about statistics. So for example, if I fully deplete all neurotransmitters that I have fully, all of them, we say, oh, probability is 1, right? We release them all. Or I can release just a portion of my neurotransmitters. So therefore, I need to introduce something that is called probability of neurotransmitter release. And what I'm trying to model here, these are actual recordings, biological cell. I'm trying to imagine how this train of spikes would result in neurotransmitter release probability. And what you see here, basically, again, I don't show the scale here, but this is the voltage. I just superimpose the voltage trace and this probability. Of course, right, they have a different units, but basically this maximum is 1. And you can see as the frequency of spikes is higher, you see it's build up, and then it kind of slows down, right, and then reaches some maximum. And then as the spike interval between spikes increases, then probability gets smaller, and then when cell goes down, it becomes 0, and vice versa. That's what you're supposed to do as a modeler, right? To figure out how the cell communicate, whether it's a fast transition from here to there, right? Whether it's a synapse is fast or it's slow. Basically, fast means is if the bump is going to be just almost synchronous with the spike here, just a little delay always, right? Because you need just a little bit of time, right? And then and then it's going to be like little bump and then goes down like this, right? Then it's fast, like it's shown here, fast. You can see really this zigzagging, these oscillations. Slow basically means that you don't see this zigzagging. It's like a smooth curve that rises up and then reaches maximum and slows down. You don't see any oscillation. Then the synapse is really slow. Okay, so, and then we have different approaches to model synaptic dynamics, synaptic kinetics. Okay, let me show you something again one more time. So you have two cells, 1 and 2. Oh, right, and then you inject a positive pulse into the blue cell, and that positive pulse injected causes a train of spikes. And then I have another electrode intracellular code from cell number 2 here, right? And what I see, its voltage goes up, okay, and then it's triggered by the full spike, and then voltage wants to come back to the negative 65 millivolts, and then the second spike in a train arrives, and you don't start from negative 65, you start slightly higher, and then even higher and even higher. As you can see, these are some kind of build-up, okay? So, which means, indicates for you that, hey, it's slow, right? It doesn't decay very fast. It's low synapse, okay? And then I see that we called summation, okay? This is called summation, accumulation, or potentiation. It's all the same terms, right? Basically, that means single spike causes positive response in a driven cell or postsynaptic cell. And when you have two spikes, the response is even higher. That's what we call accumulation or potentiation, okay? So in contrast, okay, if okay, there are two options, right? For example, I apply a stronger pulse to the blue cell, right? So I have more spikes, and meanwhile I record from the green cell, from the cell number 2, and I see that voltage. I mean, this is unrealistic. I was just trying to make it look like that it is basically you have these little negative pulses here that are directly correlated with the pulses spikes in a blue cell, which basically means that you say, the negative pulses means inhibition, plus they correlate well with the spikes, which means they're fast. Communication between them is fast. But the other option that is available, right? What if you see something like this, right? Negative 65, and then voltage goes down, so little zigzagging, then goes to saturation, and then pulse gone, and then the cell from, say, negative 75 millivolts goes back to negative 65 slowly. Okay, so we say, oh, that synapse is slow. Okay, and we treat synapse not just a direct messenger, but it's a low pass filter. Okay, and what you see on the right is a neurophysiological de facto proof of what I just said. So what happens here? This is from a SWIM CPG, so it's a circuit. Okay, this guy, Akira applies a train of pulses to this guy, right? And that train of pulses triggers action potentials in this cell. SI stands for swimming interneuron. Swimming because it's a swim generator, and interneuron basically means it's a part of the neural circuit. That's why it's called interneuron. So, and these causes train of spikes here. So you know the frequency. This is roughly 5 hertz. And then you measure what's going on in a cell on the left, right? And you can see that it's a zigzagging, right? And then as time progresses, right, this zigzagging kind of thing makes the cell more hyperpolarized, right? Like that. Okay, you say, it has accumulation or summation, right? And that synapse is very likely is going to be inhibitory. Meanwhile, you look at responses in another cell Si3 here, and spike train in cell Si2 right causes in the cell Si3 left, it causes this thing, right? Zigzagging like that. Okay, and now also they are very similar. They have the same frequency as the spike train in a presynaptic cell. And then you have two options. You can say, yeah, it looks like I have either excitatory, this synapse is either excitatory, or it can be what's called electric gap junction. We're going to talk about this. Gap junction basically means that they're not connected by an electrode, right? Like a silver wire. That means somas bodies of two neurons, they touch each other. Okay, so it's a direct gap junction. That's how it's called in neurophysiology, or electrical synapse. But you can't tell for sure. So you try another experiment, right? This time you you increase the frequency of stimulation in a cell number two, this cell, right? Now it's 10 hertz. You can see the higher frequency of spikes in the cell, and then you see what it causes in this guy, and you can see it here. Right? It's still zigzagging, but the spike frequency is so high, it doesn't have, the cell number two on the left, it doesn't have time to respond to this zigzagging, right? It just it becomes hyperpolarized very quickly, then it reaches certain saturation, right? It doesn't go any further down. And when the train's gone, it recovers. So you can basically tell in exponential function, G-league, what that G-league number should be, right? You can use this information to evaluate, to estimate the parameters of your model. Meanwhile, you look at these guys, and what do you see? You see that high frequency causes this potentiation first, but then it goes down, right? You still have the zigzagging, which means either this synapse is very fast and excitatory, or it's just electric synapse, right? And that's, I mean, again, you have to have experience to figure this out, right? Because it doesn't have any real potentiation, right? Just a little bit of moving up, but then slow inhibition kicks in and it causes hyperpolarization in a cell, right? That's how you derive the conclusion that very likely the connection between this cell and that cell includes slow inhibition and fast electric. Okay, to confirm that, what you can do, you can apply the same train to cell number 3 to see the response in cell in the blue cell on the right. And if it's going to be similar like that, you would say that, oh, that's very likely it's a gap junction and it's a two-way connection. Because again, inhibitory synapses, they have, or chemical synapses, they have threshold, and they are one-way connected. Okay, so, and the question that you should ask yourself or me, right, why there is no feedback from this cell back onto that one, right? And the point is that, remember I mentioned curare. Curare is a neuroblocker. Remember that? Curare neuroblocker. So basically what curare does, okay, when you have this preparation in the petri dish, you just drop a little bit of curare between the cells, and curare will stop spiking in the cell. Stop spiking, right? It's going to block what's called sodium channels, and that means that the cell will not generate spikes, which means it can't communicate to cell on the right, which means you block this connectivity. Okay, and just a little bit of, I mean, again, so the point is that it's a bad example, but nevertheless, if somebody is bitten by a poisonous snake, right, it doesn't directly kill the target. What snake does, right, it has a venom. The venom goes into the blood, and then that blood goes to the brain, or it goes, right, and then it penetrates, say, into the CPG, right, into the circuit, and blocks this connection, which means cell neurons cannot communicate. It destroys the rhythmic patterns generated by the circuit, which means in humans and even in animals, right, it means you can't breathe anymore. Okay, breathing is also controlled by neural circuits, right? So there is a certain periodicity, right, and there is a connectivity between, say, your pulse, blood circulation, and breathing. The higher breathing, the higher the pulse, and vice versa, right? There's another well-known example of neuroblocker. It's called TTX. Do you know, if you heard of it's called puff fish in Japan. So Japan cuisine is well known for the seafood, right? And Japanese are crazy about seafood, right? I'm pretty sure that everybody's crazy. And then they have a fish. It's called puff fish. So, but puff means basically, or it's a fish ball, right? And in a case of emergency, right, it becomes like a ball, right? So which means the predator, the other fish, can't just eat it up, right? Can't catch it because just the size of the mouth is not good enough. It doesn't fit into the mouth. But the point is that fish, it's other poisonous fish, and typically in Japan there are roughly about 20 little cases every year when professional chefs who are licensed to cook that fish do something wrong, right? For example, they do the wrong preparation of the fish. They don't remove, say, liver, because again, female fish is different from male fish, so they mess things up, right? And in male fish, liver is a delicacy, right? In a female fish, it's a source of venom, right? So you got my point. And that neuroblocker, it's a chemical name, right? But again, the abbreviation is TTX. So basically, when TTX gets into the human, you eat the fish, right? It goes in the stomach, then it gets into the bloodstream, right? And then it goes into the brain. What happens is that it breaks the connection between your respiratory network, right? It controls your breathing and the blood circulation, right? And after that, you can't breathe regularly, right? You start breathing like abnormally, right? Or stop breathing at all, right? And that basically means for human, it means death. So, and then there is no antidote for TTX, right? For certain snakes, we do know that there are antidotes, but there are other kind of neuroblockers, like in a jellyfish, medusa in Russian, right? Jellyfish. They have, that's how they catch up on fish, right? When the fish comes, is the neighborhood, right? It releases those, it basically touches with the testicles, right? And then it gets glued to the fish, and then the venom penetrates into the fish and make it quiescent, right? And then jellyfish, that's how it feeds on the prey. So it's used in the nature all the times, right? So Akira, the neurophysiologist, right, he uses all kind of neuroblockers to remove the connectivity between the cell, and then after that, just using just regular salt water or just seawater, he washes out the neuroblocker and restores the connectivity in the cell. I mean, this is one of the interesting. I mean, this is another interesting story, right? Then you can create that synapse using the computer. So bottom line is, if you have a good equations to model synaptic interaction, you can use these spikes to generate synaptic current and inject it into the other cell. We'll talk about this maybe later one day. But let's go back to synaptic interaction. Okay, so that's what I was trying to tell you, right? And as I said, you're welcome to watch some movies online, right, and read about neurotransmission. Okay, so you have a postsynaptic cell generate action potential with every spike, and again, spikes are very light, meaning that cell doesn't spend much energy to generate spikes. They should be very, very cheap or light. Okay, and basically with every spike, there is a change in the calcium concentration, right? And basically these guys shown little dots over here, right? Little circles. They are neurotransmitters, and they are released from postsynaptic cell. Okay, it's going to take some time to travel to the nearby cell like this, right? Then a knock on the door. Basically knocking on the door opens up these gates, right? They bind with the certain proteins, right? Basically that control the gates, right? Gates open, and if you've got something like this, right, negative response, this basically means that either negative cells gets inside or positive cells positively charged cells, like as I said, potassium, they move outside, and that causes this IPSP, inhibitory postsynaptic potentiation. If it's positive, it could be the other way around, but nobody knows really, right, whether it's due to positive effect, positive ions rushing out, or negative ions rushing in. So that's what I was trying to say, that these guys are always there, right? They're always reserved in what's called synaptic vesicle, right? And then as calcium changes, right, it opens up this channel, right? And then they move over here, go into this receptor. Receptor basically means this controls this opening, closing of the gates, right? It causes secondary reaction. Okay, so it's interesting, but it's like a chain reaction. So where we are at the slide 50 now, right? Okay, so what we need to do, so we need to model how the cell releases neurotransmitters, right? And there are tons of millions of these cells, right? So we can't describe every neurotransmitter, so we have to use some kind of statistics, right? So we use probability. And probability basically means that it's either 0 or 1, you release or not, right? Or release partially some neurotransmitters, right? So it's like a population dynamics, math biology modeling kind of stuff, right? So, okay. And also there is a kind of sporadic release of neurotransmitters, but again, it's probability around like 0.01, right? So it's not really a stated neurotransmitter release. So now we have to introduce, again, a few numbers. So let's say the cell, it has a rest potential -65. To generate a spike, the voltage should move above -45, maybe -30 millivolts. And then we need another level shown by this orange curve, and this is called synaptic threshold. So it can be -45 too, right? It should be slightly higher than the threshold for the action potentials. Okay, let's say -30. It doesn't really matter. It could be, but it's got to be somewhere between here and the maximum of spike, which is maybe 40 millivolts, could be 50, it could be 20 millivolts. In some cells, it's -10 millivolts. It's got to be somewhere in between. And the idea is the following, right? So when voltage in a presynaptic cell below synaptic threshold, the cell doesn't release any neurotransmitters. Probability is 0. When it becomes greater than synaptic threshold, we say probability is 1. I mean, we can always put a weight, like scale this down should be between 1, but it can't be more than 1. It could be 0.5. It doesn't really matter. So one option, we can say, we can use if, right? Again, if below 0, if above, which means probability is 1. And this brings us what is known as a Heaviside function or a step function. So which basically means that if you have an action potential, right, then every spike generate a complete neurotransmitter release. But it's not true, right? Because otherwise, you're going to release all neurotransmitters at the same time. So and then the second spike arrives and you have nothing to release, right? So this number again is just a construction, right? Some portion of neurotransmitter you release. So it doesn't have to be 1, but it's just for easy to say just 1. Okay, so again, this is voltage, this is probability. If voltage is below synaptic threshold, nothing, right? In case of the step function, Heaviside function. If voltage is above, you release everything, whatever you've got. So, but then the problem is that you have to use if, right? And as soon as you use if you want to model it, right, then you have to put if in your code, which is kind of annoying. So instead, we can use a smooth continuous approximation of the step function. Okay, this is called logistic or sigmoidal function, like this. Sometimes it's called, I think it's logistic function. So it's got to be a curve like this, right? Okay, so and mathematicians have 2 functions in reserve for these. For first, you can use arctan, right? Arctan between -pi/2, pi/2. If you add pi/2, it becomes it range now from 0 to pi, and then that's why you divide by pi, and therefore this function now is between 0 and 1. Yeah, you can do that, but it's kind of used to trigonometric function, but there is a better approach, and this you can use exponential function, but there is always a correlation between them. And the way you do this, right, you introduce, okay, if you want to model this and you don't want to use differential equations, I use algebraic equation, you use exponential function like that. Look at this. 1 and then divided. Exponential function never vanishes, right? So, but then if voltage is low here, okay, and then you put a minus there, so what happens is, if this expression, right, if this expression is negative, right, because voltage is less than synaptic threshold, and you put a number k, say 100, right, or 50 or 10, so therefore you're going to get something like e to the 10. E to the 10 or e to the 100 is a large number, and because of it's a denominator, that means as 1 over, say, large number is going to be basically 0, right? But if exponential function tends to 0, you get rid of this term, right? And therefore you have 1 over 1, and therefore you have probability 1. And this point in the middle, right, is when voltage equals synaptic threshold, and that probability basically, so this equals to, argument is 0, and then you have 1 over 1 plus 1, which is 1/2. Probability 1/2. Actually, Hodgkin and Huxley, they use these functions all the times, right? This is called sigmoidal or functions or logistic functions, right? Either way is good. So that's what we're going to use to describe our synaptic interaction. And that number k basically tells you the slope here, right? If you make k very large, just say 100, right, it's going to look basically like the step function. If you decrease it, right, and keep still make it say 1 or 2, you can control how smooth this sigmoidal function is. Okay, where we are next. Okay, now let me just talk about. I mentioned a couple times channels open, channel close, and let me draw a parallel that can kind of be helpful for you to understand. Okay. So imagine this is the room you're sitting at, right? Look, I don't know whether you're home or somewhere else, look back at the door in the room, okay? And you may have two kinds of doors, right? So if you want to leave the room, exit the room, you have to push the door out, right? So it's called outward. Or on the contrary, right, to open the door, you have to pull it. So the door, the entrance door in your house, okay, when it was built from the very beginning, right, when you enter the house, you always have to push it, right? And the reason is, I mean, nowadays, because again, I do know that in Russia, almost everyone has a metal door, right? To open it, you have to, from outside, you have to pull it. And that contradicts, again, the fireman would be mad, right? Because, again, the idea is that if you, in case of emergency, and you have to get in, you have to kick it out, right, to get in. If you have to pull it, right, then you're going to waste the time. But it's a reality, again, in Russia, that's how the doors are made nowadays, right? Again, it contradicts all common sense. But bottom line is, okay, so let's look at this, right? Example, when you raise the, say, atmospheric pressure in the room, right? You move it up, you raise the pressure, what's going to happen to the door? Door is going to open, right? Whereas in this case, if you raise the pressure inside the room, you're going to shut the pressure, raising pressure is going to shut down the door, correct? Okay, so imagine a voltage. Okay, so this is a cell, and the voltage inside the cell, right, goes up, and then the door opens. Whereas in this case, voltage goes up, the door shuts down. Okay, how quickly it shuts down, opens up, it depends how heavy that door is, right? Okay, the door could, if it's light, it opens up almost immediately. But if it's heavy and solid, it's going to take time to open the door, which basically means that that gate is either slow or it's fast, and basically what I'm showing here, this is the pressure on the right hand side, and in this case pressure goes up and the door opens, right? And therefore it's fully open and you can leave, or ions can leave the interior of the cell, right? Whereas in this case, when the pressure builds up, the door shuts down, right? Which means you can't leave the room anymore, right? Which means your probability is 0. Okay, this is called the pin curve. It's called activation curve because it's a monotonic increasing, whereas that curve is called inactivation curve. On the contrary, the door shuts down, right, if the pressure builds up. So it's the same story with the cell, right? If voltage goes up, the door can open, or if the voltage goes up, the door can the door shuts down, right? So, and now you can't leave. So there are different kinds of doors, okay? So that's what I was trying to kind of to explain to you, right? When the voltage goes up here, then potassium around this value, right? Potassium channel opens up and potassium ions, they're rushed out, right? And then voltage goes down. On the contrary, right here, when the specific gate for the sodium ions opens up, then sodium moves in the opposite direction, right? It moves inside into the cell, right? Which means, and it's positively charged, that's why spike voltage goes up, right? So it's the same positive ions, but different mechanism. Okay, sodium makes it go up, potassium rushing out makes the voltage goes down, and then there is recovery period. However, the story is not that simple, right? What if you have not just one door, one gate, but you have two gates, and they act independently, so there is no connection. So therefore, you have two probabilities, right? You have a probability S1 for this one, and you have a probability S2, and they are totally independent. And the question whether the door is open or closed depends on because they're independent, right? Depends on the product of these probabilities, right? Because one gate can be fully open, but if the other one is fully closed, then the gate is going to be closed, right? It's going to be 0. So they kind of act independently, right? And in our case, it's always true, right? So we call this, the probabilities are voltage dependent. And we're going to talk about more and more when we introduce Hodgkin-Huxley formalism. That's basically what Hodgkin-Huxley proposed. They said that the current that flows through the gate, this channel, right? What is that? It depends on how wide the gate is, okay? So which means maximal conductance, how wide it is. Difference between potentials outside and inside, okay, divided by the resistance, but we don't have resistance, we have basically wideness, it's maximal conductance, plus these probabilities, okay, that have that unit list, right, which means you have this idea of what the current is. So I have this figure twice, okay. So how much time? Okay, we are good. Okay, so let's talk about neurotransmitter rate models. Okay, so one option, we use the sigmoidal function, okay, to tell what probability is going to be. As I said, if voltage is above synaptic threshold, okay, then probability. So I'm gonna use this pin curve. Okay, so this is synaptic threshold. Probability is 0 if you're below. Okay, below the synaptic threshold, or otherwise you're gonna release neurotransmitters with certain probability, positive probability. Okay, and that's how a sigmoidal function works. And this is again it's a local jargon. If you don't have differential equations and using just algebraic equation like that, this is called FTM. It stands for fast threshold modulation. And this function, again, this sigmoidal function like this, in the local jargon is called F infinity. F infinity basically means, okay, I'll show this picture again. Infinity means at infinite values of large values of the argument. Infinity, it's either 1 or 0. Again, it's just a jargon, but get used to that. It's in all notations when you use differential equation, they use this F infinity. It's a voltage it's a function of voltage. Again, this is just algebraic equation. So again, spike, okay, spike in threshold around here. It's about to, you see, it wants to generate a spike. Then another level could be negative 20 millivolts, could be just slightly above this blue line. And then probability become 1. Okay, if I increase synaptic threshold, I have less time or more time to generate action potential. Is basically this probability could be a wider. Okay, this pulse can be wider or it could be narrower. It depends on how thick or thin the spike is, right? But bottom line is that if you generate action potential, several spikes, that FTM response, the probability is going to look like this. So it's going to be like a action potential release, okay, high frequency, high release, right? But you see, it's faster spikes, right? Basically, you generate these little pulses like that. Okay. However, the interesting part is more interesting part is another kind of dynamical synapse, right? And then you have, as soon as you have some time involved, right, then you got to use differential equations, because S is going to be not just 0, 1, jump back and forth, but it has some kind of dynamics involved. Okay, but again, this FTM is often used for fast synapses like excitatory synapses, right? But sometimes there are cases, multiple cases, when you have to take into account synaptic dynamics more accurately. And again, it's very, very important. So again, the same story, right? In a presynaptic cell, I have a spike, and then I also record from postsynaptic cell, green one, and I see something like this, right? You see, it's either EPSP, again, E stands for excitatory, postsynaptic potentiation, or I will see that negative bump, which means inhibitory postsynaptic potentiation. So I need something like this, right? And then I recently talked to John Rinzel about who was first, who introduced these models, right? And that basically was his biological advisor. He noticed that we need something like this, right? Like, you see, first of all, you have a raise in probability, and then it decays, right? And such a functions in mathematics, right? For example, if you multiply e to the negative, that gives you the decay by t or t squared, you're going to get initial raise, and then it's going to get, and then exponential function kicks in and gives you that delay as time increases. Okay, this is called alpha functions, right? They've been around for multiple times. For example, if you consider quadratic equation like a pendulum, and when you have a double roots, then that's the solution, how it's going to look like. It's called overdamped pendulum. So it looks like a fin of a shark, right? First of all, raise and then decrease. And you see this part it's kind of smooth version of that black line, right? Which gives you basically given by this. But of course, derivative at this point, the function is not. I mean, again, it's differentiable at this point, so it's going to look like more like that. And then basically what Hodgkin and Huxley proposed. Okay, they said, and we're going to talk about this more and more. Let's introduce differential equation to model neurotransmitter rate. So we need this part. Okay, so if you look at the equation, it's also differential equation of the first order. So sometimes this is called alpha synapse, or sometimes people call it alpha beta synapse, because you have two constants. So this term, if you ignore this for a moment, right? Just look at this. It's exponential function that give you that decay, right? And the rate of decay is controlled by beta. So if you make beta smaller, right, then it's going to be slow decay. If you increase this value, it's going to get high decay, right? I mean, faster decay. And this part, you're using basically FTM here, this F infinity function. And this F infinity function is either 0 or 1. Again, if you're below a synaptic threshold, F infinity function is 0. So you don't have that term. Everything goes to the steady state 0. But as soon as you've got synapse, as soon as spike is generated, right, then this function becomes basically 1. Okay. And what happens is that S will start increasing like integrating fire, right? It wants to get to some level over here. What is that level? So we put F infinity, we make it 1, and we want to find where it wants to converge this red curve over here, right? When it bends. So we nullify the S prime, right, and look at the equations on the right hand side. And what we've got, the steady state is going to be basically alpha plus alpha per beta. If beta is small, right, it's basically 1. So this S wants to go to the value of 1, and then spikes is gone, and then it doesn't reach 1, right? It decays due to this term, okay? And then you go to something like this, right? And by changing alpha and beta, you can control the initial slope over here, or you control raising slope, or you can control the delay. Not delay, I'm sorry, the decay. But also, you can call this delay if you want. Okay, so let's look at the 2 examples, right? So we use this equation, right? This is called alpha synapse, okay? And that's what we have. So suppose we have it, we apply a large pulse to our synapse, or sorry, neuron, integrating fire neuron, and we got 4 spikes from it, right? Only. So, and then we change alpha and beta. Okay, alpha is like this, right? And small beta, which means it wants to decay, but it decays slowly. And then the second spike arrives, right? And it goes up again, and then it wants to decay. Then this spike arrives, right? As you can see, it has this, looks like a dragon, right? Okay, but if the pulse was slower, or we can change this beta value, right, and make it larger, then it decays faster. And as you can see, it doesn't have this accumulation, which means that synaptic dynamics is nonlinear, right? Which means again, if I plot on this diagram, I plot spike frequency, and here I plot basically the area covered by this brown curve, right? So I need some find to average value. This green part is the average of area underneath this curve, right? So, and if I plot it as a function of spike frequency, I have options. By changing, say, I fix alpha, I don't want to change alpha, just fix it, right, and change beta. If beta is very small, which means it's going to accumulate very quickly, right? It's going to build up very quickly. And then you'll see a curve something like this, right? Which our average spike rate, or not spike rate, neurotransmitter release rate depends on the frequency. Or it can be somewhat like that, right? Which means clearly it's going to increase, right? Because it's going to be always increasing function of the spike of spike frequency, but the shape could be different. And what I need something like this, right? For my case, right? Meaning that that synapse, when spike frequency is low, synapse basically it's there, but it's very weak. And then when spike frequency is really, really high, right? Okay, define what high means, but again, we're going to talk about specific examples. Say this can be 15 hertz. Then it gets to saturation. It can't increase forever. It must get to the saturation, releasing all neurotransmitters, deplete them fully, right? Okay, and then this is what's called a working range. And as you can see, this looks like a transistor. And people from control theory, they like these functions a lot. This is called working range, okay? So where the response of the system is expectable, right? And you can predict what's going to happen, okay? In this case, the synapse basically is very slow. It immediately gives you the big response, right? It doesn't change depending on the spike frequency, which means it's not really a nonlinear function of the spike frequency, right? It grows very quickly, right? You can always say like vertical and then flat, right? These could be just almost linear, right? Increase, which is not bad at all, but nonlinear dependence like that is the most effective. So which means you have a cellular dynamics. You do know that number of spikes depends on a stimulus, and now you have a slow synaptic dynamics or fast synaptic dynamics, right? And now you can bond things together. Now you can combine things together. Okay, and this is, I showed you this diagram before, and what's shown here, right? That's a file is called alpha synapse and FTM. Okay, that file, and basically what I do, I show here. Okay, this is my integrating file. I can change the number of spikes by changing this input. And what I model here, this is FTM, basically. I want to model the probability of neurotransmitter release. You see it's 0, and as soon as it generates the spikes, I have basically I have a vertical pulse, and the reason I put the dots here, basically because it shows that from probability of 1, it goes straight to 0 immediately, okay, which means it's very fast. So it's like a pulse. Whereas when I use alpha synapse, I have a build-up and then decay back to 0 because pulse is gone, and then I have another one. So basically, I click here. What the file is that? It's called alpha synapse FTM. Yeah, this one. Okay, when I click, it's exactly the picture that I've got. So the only thing I would like to do here is when I plotted FTM, I'm going to remove this, okay, and do it again. And now you can see that probability is 0, jumps to 1, immediately with spike, and then back to 0. If I change, and now I have synaptic threshold, it's -2, and -1 is spike threshold. So it's kind of funny, right? It should be the other way around. Synaptic threshold, it should be -25. Goodness me, let's do that. Okay. And now you don't even see here, right? Okay. Let me go back to that. I make it just equal, right? Because I just wanted to see the build up. We see it builds up very quickly. So if I remove this part, okay, so I'm going to connect the dots, you will see how it works, right? This spike gives you a very fast build up. It never reaches the value of 1, but it doesn't really matter, and then slows down, right? But if I increase the frequency, if I increase the drive, right, so I should generate more spikes. Click, I generate more spikes, okay? FTM is going to give you probabilities like these, right? But it doesn't give you build up, right? It doesn't give you accumulation, where in this case, you can see I have this accumulation. But also, if I, let me see, where is alpha? Okay, if I make better, keep alpha constant, make it 0.1, and run it again, you see I have a build up. So the question is, on average, what is stronger? Here, this and that, right? If I don't assign weights. This is very quick but instantaneous. And the area is very small, whereas this area is, on average, right, is kind of large, which means this synapse at the higher frequency stronger than FTM. So if I move it back to 6, then, okay, I still have a build-up. If I make it 5, hopefully it's going to 5 still, right? I still have a build-up, and this is slightly unrealistic, which means this constant beta is way too small. So I have to increase it to make sure that spike, after single spike, the neurotransmitter release vanishes, right? Maybe even this not good enough, so maybe I should increase it and make it as it used to be, like that. So it goes like this. I increase the spike frequency, I'm just make it 15, and I should have a build-up. Okay, so on average, my level is around here, right? Whereas this average is very, very small compared to that one. So which means synaptic strength is a function of the frequency of the cell. And then I have this file, let me see, alpha synapse frequency, okay. Info synapse alpha, I right click this, okay, and then let me run it. Okay, here I'm increasing the drive, just showing the FTM, the green line, and then alpha synapse build up as time progression, right? So as you can see, the average level for the green is here, over there, and for the alpha synapse, the average value is around here, which means the synapse gets way stronger than FTM if I would like to model it like that. So that's a file. It's called integrating file alpha synapse frequency depending on the drive, right? Okay, so I'm going to close this. This is exactly what I was looking. The average probability as a function of frequency. Frequency goes up, FTM doesn't change, almost straight line, right? And these guys, they have nothing before 5 hertz, right? This alpha goes quicker, and this is the model that we developed specifically for our purposes, right? I don't want to discuss it today. Okay, so I close that. We still have time, and okay, guess what? Let me just announce something, and then we're gonna stop. So I need to. Okay, this is PowerPoint clearly, right? Okay, so I'll just show you a few more examples. Okay, and that is gonna be it for today. So again, these are biological recordings. Okay, so I borrowed them from Akira. I have the data, right? And that's what he can see when he poke electrodes in a cells. And he does know that this guy talked to this guy. So whenever this guy spikes, this one is quiescent, right? And then they reveals the orders. And you can see, for example, this inhibition, right? When the spike frequency drop down, the inhibition weakens. This guy wants to wake up, but it doesn't have energy. So we can measure the frequencies, right? And this is my modeling studies. I'm trying to model how the blue cell, this dark blue cell, talks to the other cell of the network, and how this red cell talks to the other cells of the network. And for me, what is important is that you can see this spike frequency is always the same here, right? Which means, I say that I model it using these alpha probabilities, right? Alpha synapses, and probability basically stays constant, right? Despite these little things, right? But when the spike frequency drops down, you see probability, it builds up a little bit, but it's not as strong as that one, right? Because the spike frequency is way, way lower. Whereas this dark blue means very, very high density release, right? Corresponding to the higher frequency. It drops down, that's why you have the sag, and then it fluctuates, but again, on average, you can kind of know how it's going to look like. And for example, this fast burst is going to give you that release. Okay, and to conclude, okay, and the next time we just we will model interaction between the cells, but imagine that you have again two cells, okay, and if the cell, say this is spike frequency in the cell, and when the cell gets positive input or excitatory input from the other cell, it should accelerate the spike frequency. It should go up, okay? So that was, and when the cell gets a negative input or inhibitory current, right, then the spike frequency should drop down, right? That's what we're trying to model. And how we model synaptic interaction. Again, this is my equation. This time, okay, when you model synaptic interaction, you always put minus here, not plus, but again, I'll explain why. And the idea is the following. So let's say if the input is positive, right, that would be a normal increase in the voltage, but then it's going to get an extra boost from excitation. It's going to grow faster. Whereas in the second case, when the cell gets inhibition, that's how it would normally grow. Inhibition delays it or slows down. And how we model this, okay? So the idea is that we know S, okay? Now we can measure synaptic current we can calibrate synaptic current by the resistance or Gmax. It's called maximal conductance. And most important part is this, okay? So for the current, you must have the voltage difference, right? Voltage here. And this is the voltage in the cell, and this is called synaptic reversal potential for the synaptic current. So let me explain. Okay, so if I want to get a positive boost, okay, so I want to make sure that this synaptic current here is going to be negative, right? Which means I'm going to get plus here. So let me see. That's my voltage, okay? If I put, for the inhibitory current, make this V seen at the level of negative 90. So therefore, what do I have? My equation, I've got minus there. G is positive because conductance. S is positive because probability. And then I have V minus this negative 90. But because negative 90 transforms into the plus here, therefore, and voltage changes between negative 65 and 40, which means this expression is always positive, which means, okay, the whole expression, synaptic current, right, acts as an inhibition, right, because of that minus. But if I shift it up here, 50, then this difference is going to be always negative, which means the whole expression always positive, which means it's going to be excitation. This current modeled by excitation, by choosing the level for the reversal potential, negative 90 below voltages of the spikes, right? You create inhibitory current, but if you move it up over here above the high level of spike, that's how you model excitation. Okay, so that's how you model. That's how you introduce synaptic currents. Again, two types. It can be a positive synaptic current, which means excitation, or negative synaptic current, which means inhibitory synapse, by choosing this level, right? In the inhibitory case, negative 90, but it doesn't really matter, negative 90, or it can be negative 70, but it's got to be below the voltage of spikes, right? Whereas in case of excitation, it's got to be either above, or on average, it's got to be over here. So to make sure that on average it works the same, right? Okay, finally, maybe, no, I think it's enough for today. I'm tired, I'm pretty sure you're tired too. Let's stop here and just remind me next time. So we have to start from this slide, and then we're going to change the, we're going to consider networks of integrated fire neurons. So again, this is a Dropbox. You're supposed to get a link. So I suggest you download the files. If I go back to the same directory over here, then if you click at lectures, then lecture number 1 is a PDF that I showed you today. We discuss all the slides, okay? And that's where we stop, right? So I want to introduce what electrical current is, but I'm run out of energy, and so I better stop here. All right, questions, guys? If you're still here.
Да, можно спросить.
Давайте.
У нас экзамен будет в виде проекта?
Угу.
А когда мы узнаем тему проекта?
В следующий раз. Я вам дам несколько заданий, и может быть одно задание. С другой стороны, одно задание вполне достаточно.
И нам что, нужно будет написать как программу?
Вам нужно будет сделать отчет, как я показал, вот предыдущие ребята делали, например, что-то подобное.
Угу.
То есть я вам скажу, что надо делать, да, файлы у вас есть, и как эксперимент, и вы просто рассказываете ваше видение, как это моделируется и прочее. Oh, I'm sorry, Victor is still here, right? So basically, I'm sorry, I'll set up what you're supposed to do, right? And then you just do your research part, and then write either a PowerPoint presentation, right? Or if you want to write something in LaTeX or Microsoft Word, it doesn't really matter. So something like a short article research study. Is it clear enough? Hello?
Ну, нам понятно.
Good. All right.
And how much lectures do we have?
How much what?
How much lectures do we have?
Look, I'm sorry, I missed it. Lectures.
Oh,
Oh, I got the lectures. So we don't have a lot, right? So today we had 2 lectures basically. Total is 8. So which means 4 days, right? It's going to be today, next Thursday, next Saturday. So we're going to meet on Saturday, and it's going to be another Thursday. So which means 4 lectures.
Okay,
oh, not 4 lectures. 4 days, 8 lectures.
I see. And when can we start our projects?
I'll let you. The problem is that I don't have your emails. Very likely, I'm going to either email the topic, I mean the themes of the project to you, Alexei, or whoever is in charge of running this show, which is, let me see, I forgot her name. Let me see, either Alexei or, let me see my emails, or Natalia Chebochko.
See the point.
Huh?
I see.
Because I don't have your emails, guys. Normally, when I I told you, right, the way it's done here, we have a platform, and on that platform, I have access to all students' emails, so I can email the whole class what the project is all about, right? At this time, I need a middleman between us.
I see. Thank you.
It is you. But again, I mean, as I said, this is today's lecture, right? I didn't have time to. I do suggest that you run these MATLAB files that I showed you today, at least to see what's going on, right? And I do suggest that you download them to your local computer, put them in the same directory the way I did it, right? So you can run them and see the outcomes. All right?
То есть у нас будет 2 недели на проекты, я так понимаю? У нас до 27-го, как бы, или как? Это же, я так понимаю, оценка за проект будет как экзаменационная идея.
Yeah. So basically, 27-го вы должны дать мне свои проекты. 27-му.
А 27 можно проект писать на русском?
Пишите, конечно.
Спасибо.
Конечно, как вам удобно.
А еще такой вопрос. Проект обязательно должен быть выполнен единолично или можно в группе делать?
Ну, желательно единолично. I mean, it's your own grade, right? It's not a group grade. Это не оценка же за группу, да? С удовольствием обсуждайте, но старайтесь не копировать друг друга и привнести оригинальность в каждый проект.
Хорошо, спасибо.
Пожалуйста. Молодые люди, я жму вам руку и прощаюсь с вами. У меня еще много на сегодня дел. У нас только лишь час дня. Всего вам доброго и до свидания.
До свидания.
До свидания.
До свидания.
Спасибо, до свидания. До свидания.
Пожалуйста, пока.